Hello. In today's lesson, we're going to discuss how we can use multiplicity and modeling in order to determine interesting things about the human genotype and make predictions about genes that we inherit. The human genome is about 3 billion base pairs long. To put that in perspective, that's about the number of seconds that make up 96 years. 96 years ago from 2015, the 18th Amendment was just being ratified to begin prohibition in the US. Humans also share about 99.9% of their DNA with other humans, so only 0.1% of DNA makes us unique. And we know how unique certain individuals can be. That leaves us with 3 million base pairs that make us unique, or about 36 days if we use the same comparison as before. That's still a lot of variation when we look at multiplicity. What multiplicity allows us to do is quickly and easily determine the total number of combinations of something. For example, ice cream. Say you're getting a triple scoop and you have 3 of your favorite flavors, chocolate, vanilla, and strawberry. How many ways are there to scoop your perfect cone? The answer is 27. Now, that's a lot of ways, but we could have come to that conclusion much quicker by using the multiplicity formula and saying that there are 3 options for the first scoop, 3 for the second, and 3 for the third. That's 3 times 3 times 3 equals 27 again. When considering the human genome, we can say each base pair in our unique DNA is a category, and the options in that category always equal 4 for the base pairs we have available: adenine, thymine, guanine, and cytosine. So instead of multiplying 4 times 4 times 4 forever, we can use the multiplicity formula again and simply express this as 4 to the 3 million. This will give us the total number of genotypically unique humans that are possible within the length and structure of our genetic code. That's 10 to the 10th to the 5th times greater than the age of the Earth, which is already 4.54 billion years old. Wow! So yeah, you'll never run into your exact clone unless you have a twin or underwent cloning procedures, in which case, congratulations, you literally just beat all the odds to find them.
Let's take a look into a more likely and more applicable case: siblings. In this case, we can safely assume that the genes are inherited from the same parents. What is the probability that you and your sibling will inherit the same exact gene? Sexual reproduction requires meiosis, a process in which a parent cell divides to produce cells with half the genetic material of the parent. Chromosomes replicate, separate, and then divide into single chromatids segregated into individual gametes. The haploid gametes from 2 individuals combine to produce a diploid zygote, you. But if there are only 2 versions of the genes between your parents and your sibling, you should have each 50% chance of receiving a particular version, right? False. Let's take a closer look at the process of meiosis. During prophase 1, DNA is exchanged between homologous chromosomes, resulting in chromosomal crossover. This process of homologous recombination is a significant source of genetic variation and can transfer or alter the genes on a chromosome. When calculating the probability of an individual receiving a particular gene from a particular parent, we must also account for the probability of recombination occurring between chromosomes prior to gamete formation. We want to know the likelihood of the crossover point occurring within the gene sequence, which would result in a new variation of the gene. The probability of recombination occurring is directly proportional to the length of the chromosomal segment. However, if the gene were to extend the entire length of the chromosome, an arbitrary length n, the maximum probability of crossover between these 2 endpoints will still only be 50%. So we know that for a gene spanning a length L, the probability of recombination within the bounds of that gene is proportional to the length relative to the length of the chromosome, and multiplied by the maximum possible frequency of 0.5. Since we are interested in the probability of you inheriting an intact gene, we take this value and subtract it from 1, and then multiply it by the factor of 0.5. That accounts for random assortment in other stages of meiosis. We now know the probability that you and your parent have the same gene. To determine if you and your sibling both share this gene, we simply square this value.
So why is this useful? Why should we care which genes we share? Many diseases are caused and regulated by specific genes in an individual's genome. If your parent or sibling has been diagnosed with a genetically linked disease, what is your risk of also carrying the gene and developing the disease sometime in the future? Imagine, for example, that your dad carries the APOE gene located on chromosome 19. Individuals with this gene are at a high risk for developing Alzheimer's. What is the probability that you or your sibling have inherited this gene, or that both of you have? Using our model and the known information about the APOE gene, we can make this prediction. APOE is 3,646 base pairs long and is located on chromosome 19, which has a total BP length of 59 million. Using these numbers, we see that due to the small size of this particular gene, the probability that the gene has remained intact is roughly 99.99%. Continuing with the calculation, we see that there is a 49.99% chance that you will also carry the APOE gene and a 24.99% chance that both you and your sibling are carriers. Knowing this information can help you and your family to prepare for the case that you would develop Alzheimer's. This same concept could be applied to other genes that are linked to diseases, allowing individuals to take preventative measures. As we continue to identify and map genes within the human genome, models such as ours will allow us to keep individuals informed about their potential risk of inheriting or sharing genes that cause certain diseases. Thanks for watching! Subscribe to our channel and join us next time for more BM Easy.